So, very good afternoon. Today we discuss that is block third means block one unit third. That is the what unit third physical and biochemical changes during the seed storage. That is block one unit three. So what are the changes during the seed storage? Means physiological activity, either biochemical activity. What are the changes occurred during the seed storage time? So during the seed storage, the seed moisture content and temperature are responsible for the seed determination. So many physiological and biochemical changes are occur in the seeds during the seed seed aging. So deteriorative changes. So deteriorative changes enhance when the seed exposure to external challenges increases and decreases the ability of the survive of seed. It is an undesirable attribute of agriculture. The annual losses due to the deterioration can be much as 20%, 25% of harvested crops. It is one of the basic reasons for low productivity. So the biochemical changes associated with the seed deterioration is the increase in leakage of biomolecule, according to Kohler in 1985. The physiology of seed deterioration is the distinct consequence from seed development and mm, germination. Losses in seed quality occur during the field weathering, harvesting, and storage. The several factors contribute to the susceptibility of seed deterioration of the seed. Okay. So, so here, what are the deterioration types? Here, that is the wait. So, deterioration types we have to discuss here. So, deterioration is the evident as the decrease in percentage of germination, while those seeds that germinate produce weak seedlings. Losses in seed quality occur during the field weathering, harvesting, and storage. The harvesting time of survive several crops depending on the it is maturity time and on the physiological maturity. Harvesting time of several crops, the harvesting storage influence the quality of seeds, germination, vigor, viability, and also storability. Physiological maturity is attained is then genotypic character, which is influenced by several environmental factors. The moisture content of physiological saturated seed is approximately 50 to 55%. Because of the high moisture content, the seed cannot be harvested commercially and adequately low to permitting mechanical harvest without causing damage of the seeds. So that is the here. What are the uh, so field conditions are rarely favorable for such storage. Seed quality is influenced numerous factors that occur in the field before harvesting and during the harvesting time. So drying process and storage, the losses are uh, rust of seed storage at high temperature and high relative humidity condition. Weathering, another type is the weathering. The seed deterioration quality, vigor, and viability due to high relative humidity and high temperature during the post maturation and pre-harvesting period is refers to the field weathering according to the Bhatia et al. In 2010. The weathering occurs the period between the attains of physiological maturity till harvesting the field. Duration caused by the weathering is the directly related to seed exposure to adverse condition. So that is the physiological quality is depending on the environmental condition preceding harvesting. So adverse environmental conditions during the seed filling and maturation result in the forced seed maturation, which is associated with the how low yield, leading to a specification decrease in the quality and and exclusive reduction in the crop productivity. After physiological maturity, if the seeds are retained on other plant seeds will deteriorate. The physiological changes in the seed may lead to formation of rigid seeds or of color seed in the purple crops. Harvested delays behind the optimum maturity, extended field exposure and intensity intensified seed deterioration. Weathering are only the low seed germination, but also increase susceptibility to mechanical damage and disease infections. So timely harvesting, avoid prolonged exposure to moisture, and it is the best means of avoiding weathering. Harvest and post-harvest deterioration seed quality is highly affected by harvesting and handling method. So deterioration in the post-harvest so comprise the um, uh, threshing and the processing, machinery and seed collection, handling, transporting, and drying during the storage. Mechanical damage, one of the major cause of seed deterioration. Very dry seeds are prone to mechanical damage and injuries. Such damage may result in the physiological damage or fracturing of the seed pods, broken seed coats, permit early entry and easy access for microflora make the seed vulnerable to fungal attack and reduce the storage potential. Cotyledons and that is the shaded and broken seeds. Large seeded varieties are more sensitive to mechanical damage than the small seeds. Okay. Large damage seeds that is very susceptible than small seeds. Storage of seeds. So sterility of seeds mainly in a genetically regulated character and influenced by the quality of seed at the time of storage and pre-storage history of the seed. Moisture content of seed or ambient relative humidity, temperature of storage environment, storage duration and biotic agents. The damage of seed during the storage is inevitable. The seed storage of seed environmental highly influences the period of seed survival. After planting of deteriorate seeds, seedling emergence may be poor or transmission of pathogens to the new crop may be occur. The low temperature and humidity result the delay the seed deteriorative process. There is by leading to prolonged viability period. Seed deterioration mechanisms. Once seed deterioration has happened, this catability process cannot be reversed. It is the sequence of event beginning with a chain of biochemical event beginning with a chain of biochemical events, predominantly membrane damage and emergent of biosynthetic reactions and then the resulting losses of various seed performance attributes, starting with the reduced germination rate, reduced field emergence, increased number of normal seedlings, and finally seed death. Viability loss result in the irreversible chemical and structural changes to cellular constants. The structural changes associated with the oxidation of reduced membrane fluidity, alternate folding of DNA, lost elastical of proteins, and increased brittleness of the cellular matrix. So, biochemical manifesting of the seed deterioration. The seed deterioration is the associated with the various cellular metabolic chemical alternation, including the chromosome aberrations and damage to the DNA and impairment of RNA proteins synthesis changes the engine and of food reserves, loss of the membrane integrity. Some of the major physiological and biochemical event of deterioration are the present we can discuss. Membrane degradation. Membrane degradation. It is an exclusively content that loss of cellular membrane integrity is one of the primary cause of loss of viability. Under harsh storage condition, loss in the membrane permeability leads to increase the leaching of seed constant and hence is loss of viability. During the seed deterioration, The membrane degrade, increase electrolyte, electrolyte leakage, decline in seed germination. Field emergency and seedling vigorous is associated with the high level of electrolytes leakage. So alternation of membrane system such as the tonoplast, plasma lemma, and endoplasmic reticulum result in diminishing the normal cell functions and energy production. The the membrane deterioration and loss of permeability occur at the early stage during the seed deterioration. Enzymes alternation. Enzymes alternation such as reduced activity of lipase, ribonulose, acid phosphate, protease, dietase, catalase, peroxidase, amylase, de- dehydrogenase, and ROS means reactive oxygen stresses. So Paradise activity decreases sustainably with the aging. Due to this, seeds became more sensitive to the effect of oxygen and free radical in the membrane. Understand many fatty acids and produce lipid peroxides products such as metabolites and lipid conjugation. Changes in the cell membrane, chemical constant. So, what are the changes are occur in the chemical compositions? So, in the deterioration, significant decrease in the protein, oil content, and total sugars, and increase in the free fatty acids, and reducing the sugars has been studied. Some of the studies include the oligosaccharide, which has been associated in the stabilizing the membrane and decrease the during the storage. Reduce metabolic activity, high relative humidity, constant deterioration, and result in the reduction of the nucleic acids with the increased storage period. Metabolic activity of seeds we grow in non-viable seeds to the viable seeds. Long-term storage decrease the ability to the nucleic acid and nucleotide. So next topic is the free radical damage. So deterioration is the partially associated to accumulation of the free radical produced by the metabolic process. Seed storage subjects the lipid to the low constant attack by the oxygen, the forming the hydrogen peroxides and other oxygenated fatty acids. Free radicals. Free radicals are the unstable and may react and damage nearby the molecules. Oxygenated fatty acids in the absence of enzymes activity in the dry seed would accumulate and damage cellular component and leads to the seed deterioration. Lipid peroxidase and free radical formation are the major cause for the deterioration of the oil seeds in the storage. Chromosome aberrations. So, chromosome aberrations, one of the changes linked with the seed agency, the aberrations of chromosome, sometimes permitted to the mutagenic effect. Some of the chromosome alternate in seeds comprise of the fragmentation, bridges, fusion, ring of formation of chromosome, and various in the nuclear size. Seed deterioration symptoms. So the seed deterioration is the inexorable and irreversible deteriorative changes in the quality of a seed. It is the adversible in their dropped performance during the germination, such as the delayed seedling emergence, slower rate of seedling growth, and development, loss of the capacity to the germination was at the final phase of duration. Process and final indication of vigor loss. So deteriorated seeds are also diminished resistance to environmental stresses during the germination and early seedling growth. Some of the symptoms of the deterioration of seeds are what here morphological changes. Some morphological changes are occurred. So their seed coat color are approximately apparently owing to oxidative reactive in the seed coat that are the enhanced under the conditions of high temperature, relative humidity, ultrastructural changes. An electron microscope examines two broad pattern of the uh, coalescence of lipids bodies and plasma extraction of from the cell wall related with the deterioration have been observed. Both of these events influence cell membrane integrity. Cell membrane changes. As a cell deteriorates, there is a capability to retain the cellular function. Uh, some consequences of membrane damage include the breaking the plasma membrane, plasma lemma structure, and it also attraction from the cell wall. Fragmented endoplasmic reticulum and locking of uh, polyribosomes, lack of the dictyosomes, lysosomes, ability to, depending on the its cytoplasmic distinction of the mitochondria and plasma. So, and the plastids, all causes of lipid polymorphism and chromosome lobe nucleus and the layers of membrane of the chromosome structure. Loss of the enzymes activity and reduced respiration. The most sensitivity test of the maturity early peroxidase and amylase cytochrome oxidative also related with the seed of deterioration. As the seed deterioration, respiration rate closely associated with the seed deterioration. Increase in the speed leaches and increase in the free fatty acid content. Increase in the leachate content when the soaked in water is often match are symptom of deterioration seeds. The technique, the leachate concentration can be method by the determine the soluble sugar contents of the leaches. The hybrid is phospholipids leads to the humidity. Methods for testing of the seed determination. So germination test. So seed germination tests that is so germination is analytical procedure to evaluate the seed germination under standardized favorable conditions are there. So standard germination test which is include the media, temperature, moisture, light intensity, and seed dormancy germination counting standard for various crop seeds. Tetrazolium test. First one is the tetrazolium test. So tetrazolium test actually is accept for the accurate the mean of estimating the seed viability. This method was developed by Professor George Locon in the early 90s. It is the quick estimation seed viability. This test is distinguished between the viable and dead tissues of embryo on the basis of their relative respiration rate in the hybrid hydrate state. Electric conductivity test at the seed deterioration process, the cell membrane became less rigid and became the more water permeable. It is allowed to cell content to leakage into the solution with the water and increase in electrical conductivity. It is possible to rapid condition of seed viability for seed loss. Vital coloring test. The principle of this method is the different coloration of live against dead tissues. When they established in the certain dyes such as the sulfuric acid, indigo caramine, and aniline dye. So these dyes stain dead tissues blue and the live tissues left over the pattern the useful to determine the viable. So enzyme activity. The test, there is a method measuring the activity, that is the lipase, amylase, and diastase, catalase, peroxidase, and dehydrogenase of invade seeds as they indicate in the viability. So what are the here summary we have to discuss again? The So during the storage, seed moisture content and temperature and also responsible for the seed deterioration. Many physiological and biochemical changes are occur in the seed during the storage aging. So deterioration is the evidence to decrease the percentage of the germination viable. These seeds that is germinate produced weak seedlings. Losses in the seed quality occur during the field weathering, harvesting, and storage. Once seed deterioration has happened, this catabolic process cannot be reversed. It is the sequence of events beginning with the chain of biochemical events, beginning with the chain of biochemical event, predominantly membrane damage and possible impairment of biosynthetic and reactions, and then the resulting losses of various seed performance attributes. So tetrazoline test, that is the extensively accepted and accurate mean of the estimation of seed viability. So that is the now we have to discuss block second, and the block second also three units are there. Unit number four, seed viability and genetic evaluation. Unit number five, that is seed marketing. Unit number six, seed treatment. Here block three also we have to discuss three units are there. That is what seed viability and genetic erosion. So now we are to discuss about the unit number four, that is introduction. So for the most seed technologist and commercial seedmen, viability means that is the seed in capability of germinating and producing normal seedlings. So viability denotes the degree to which a seed is alive, metabolically active, and possesses enzymes capable to the catalyzing and metabolic reactions are needed to germinate. This is the this meaning is the deals with the tissue's viability as well as the viability of entire seed. Seed viability is the probably highest at the time of physiological maturity through environmental conditions on the permanent plant may not be permanent germination. After physiological maturity, the viability of seed to gradually decline. Some factors affecting seed viability, cultivars and harvest viability. So different cultivars and harvest of the species show different viability characteristic under the same storage conditions. Significant difference in viability period exists between the genotype with the species. So initial viability has been established well matured seed initial germinal capacity can the longer period meeting capacity. Pre-harvest so the mechanical injuries infected with harvesting can severely reduce the viability of some seeds. So the presence or absence of surrounding structure of the seed during the storage itself can influence validity. So injuries, those two vital part of the embryonic axis usually bring about of more rapidly loss of the viability during the storage than the that is the injuries that is located. Tetrazoleum test for the seed viability and seed witherers. So germination test is the best indication for the potential the seed lost would emerge under the field condition. However, it takes from the 2 weeks and in some cases even months to complete the this necessitates development of rapid method for estimating the germination behavior of the seeds, out of which tetrazoleum test, commonly known as TZ test, seed viability, have therefore been developed. Such fun is quick estimation of seed germinability. This test is very useful to process handling, storage, and marketing losses inquiries of such in the short time. In testing, you determine seed loss vigorous rating of the seed losses, supplementing the germination test result and diagnosis of the cause of seed deterioration. What are the advantages of petroleum test? Retest. So the seed viability can be estimated using tetrazoleum test is much shorter time and compared to germination test. Therefore, this test is quite sustainable for the rapid evaluation of the large number of seed. The technique can be used even with the determines its the seedling growth is eventually low so and according to the seed can be determined with the performance test to dormancy. Since tetrazoleum test is required a close examination of seeds, seed amylase can be often to get an idea to uh, reasons for poor viability of seeds. That since dicot seeds need not to damage during the tetralogy test, some seeds could be replaced for further investigation. What are the disadvantages of tetralogy test? Evaluation of staining of patterns and their interpretation in the terms of seed viability is the difficult, requires and considerable experience in the painting of work. The total work involves in the total test will require a greater number and manpower and the tetrazolam alone is unable to differentiate between the dominant and non-dominant seeds. So it is not possible to direct the process of seeds of microorganisms harmful to the germination of seed. Injury caused to seed by microorganisms, fungicides, fumigation may not be get the. So next one is the genetic erosion. Genetic erosion. Genetic erosion. The term genetic erosion describes the process of displacement of old and indigenous land races and varieties by new high yielding varieties, which is the equated to loss of the genes. First observation regarded the loss of local variation from farmers' field in the terms of their number and their specific genetic variation and specific allele in gene complexes. There are three different views and methods of quantify genetic erosion. It can be as the genetic erosion is absolute loss of crop varieties or alleles. The use of an absolute loss of evidence of genetic erosion ignores the dynamic nature of the farming system and population genetic processes. This approach only looks at what has been lost and not at the what is the replaced with this lost material. So the genetic erosion has reduction in the richness. A reduction in the richness mm, is the better indicator of genetic erosion as the just recognize the dynamic in the system. So reduction is the richness is the always reduction in the richness. Loss may be compensated by the genetic erosion is reduced. The genetic erosion, genetic erosion is the reduction in the evenness. So the reduction is evenness originated from the diversity indicates used in the vegetation, ecological and population genetics. The diversity is measured using the frequencies of allele with the group of genotype or using the productivity area of land races, cultivar or crop species and region. Diversity level are allowed due to the increased dominance of the single or smaller number of crop species. Genotypes are alleles, even though alleles are varieties are not necessary lost. So using evenness, rare varieties, rare alleles contributes little to the diversity. The risk of losing alleles varieties are higher when the distribution are very frequent. So using the events on the measure for genetic evaluation offer the opportunity to take action before the reduced diversity result in the absolute loss and reduced richness. Um, that is the So analysis of genetic erosion in the different level of integration. So loss of the cross species. Genetic erosion as reflected in the assemblage of crops used in agriculture was also recognized by Arlan as he started. So varietal erosion. Genetic erosion as a loss of varieties sometimes described as varietal erosion. The focus of most of these studies has been the transition stages, which is the land races are replaced by the modern cultivars. The two approaches to quantity of the loss of land races have been used. The first approach is comparison of the number of land races or botanical varieties found in the area during the collection mission of different times. A possibility problem with this approach is that more intensive survive may yield more land race. It may be difficult to copy the approach of the original collection mission. A second approach is the interviewing the farms about the land race for the growth of area. So genetic creation reflected in the pedigree. So pedigree studies have been used in the attempt to overcome some of the problems in the assessing the diversity based on the varieties. Using the information on ancestors, these studies estimated the determinate destruction of cultivars and the except to which the old land races, the present the degree of the modern cultivars. So allelic erosion, the development of molecular technique is in the last decades has made it is possible to study genetic erosion at the level of the alleles. The drawback of the studies and genetic erosion at the level of the varieties are using the pedigree information overcome by looking into the more detail at the genetic makeup of the genotypes. Allelic richness is important for the survival of species. A significant loss of alleles can affect the evolutionary potential for the modernization bottleneck. So replacement of land access with the modern cultivar, the first stage of the modernization. So bottleneck possible lead to the genetic erosion in the replacement of land races by modern cultivars. The replacement of land races with the modern cultivars is a gradual process and the length of the transmission period will vary much between crops and regions in developing countries. The replacement of landrace is currently in progress, while in the Northern America and many European countries, for many crop landraces have been absent and only modern cultivars are grown by the farmers. The first cultivar introduced in the area will not immediately displace landraces and therefore is likely that the total diversity will be initially shown in the increase. So, what is the summary here? The viability means that the seed is capable of germinating and producing the normal seedlings. Germination test is the best indicate the potential of seed stars to emerge under field conditions. It is take the from day to weeds in some cases even more months to complete. This necessitated development of the rapid method for the estimating the germination behavior of the seeds, out of which tetrazoleum test, commonly known as the t-test for seed viability, have therefore been developed to furnish a quick estimation of seeds germinability. The test is very useful in processing, handling, storage, and marketing large quantities of seed times. Testing determines seed loss, vigorous rating of the seeds loss, supplementing the germinating, result in the diagnosis of cause of seed deterioration. The term deterioration describes the process of displacement old and indigenous land races and varieties are new and high yielding varieties, which is equated to loss of the genes. The first observation is reported varieties from the farmer's field to term of their number, not their intraspecific genetic variation or specific allele or the gene complex. Genetic variation is reflected. It is the assemblage of the crops used in the agriculture, but are also recognized as Ireland. It is started with the providing supporting data. Genetic erosion is the loss of varieties, plant races, and cultivars. Sometimes is described as mm, varietal erosion. So that is the fifth unit is the seed marketing. So seed marketing, now we discuss about the introduction. Seed marketing. Yeah. So seed marketing is one of the most vital component of seed technology on the depending on the size and scope of the seed industry. Broadly, it is includes such activity as production and processing, storage, quality control, and marketing of seeds. In the narrow sense, however, the seed marketing is referred to actual acquisition and the selling of packed seeds, intermediate storage and delivery and sales promotional activity. It is the present context. Our discussion is limited to the seed marketing in the narrow sense. So seed marketing is the companies following the aspects of the damage for facts and marketing structure, arrangements and storage of seeds. And fourth aspect, the sales promotional activity. Fifth one is the post-sales service. And finally, economic seed production and seed uh, pricing. Okay. So this is the some class for the seed deterioration and the seed viability and the seed viability tests. That is the seed marketing. Some of the topics we have to discuss. Okay. So tomorrow we will discuss about the remaining uh, class.